So I'm just recording this now and I'm just going to share my screen. Okay, I think there we go. So I think that is shared. So I'm not sure how much of this PowerPoint I'll actually get to. Like I said, I want to spend some time talking about respiration and photosynthesis and talking about those reactions and some of the things that are important to know. So just a reminder about the midterm in general. This is kind of the plan. Like I said, I haven't written it yet. I may tweak things around a little bit. I got to see what I did in previous years. I think maybe I had 2 long answers before, so I'm not sure if I'm going to follow that formula or not. Writing exams online is a little bit of a different experience, I think, and for all of us. And just a reminder, we're going to be using the lockdown browser. So I'm just writing a note to myself to remember to put up the sample quiz so that people can make sure their technology is working. Okay. So this is a reminder of the topics that we covered. So the big ones are the last 3 because all of those were about 2 lectures each. So I want to spend some time right now on respiration and photosynthesis. So this is an important slide. This shows respiration and all of the reactions except for fermentation. And so what I want to do is just spend a few minutes kind of reviewing all these reactions here. So take a look at this. Remember, there were 3 things that I talked about, which are kind of almost all represented on this slide, right? So we've got the carbon. I'm just going to use a C for carbon. Not enough room to write it there. We've got electron carriers represented up here. And we have our ATP represented down here. So what I want to do is kind of just overview these reactions. The nice thing about this particular diagram is it's showing them all, and it's also showing their locations. So glycolysis happens in the cytoplasm. So let's start there with glycolysis. And like I said, we can kind of think about this in terms of those 3 items, right? So let's take a look at the overview. So glycolysis, we start off with glucose. There's my carbon. I don't know how to change the pen color in PowerPoint, so it's all going to be in red apparently. And then, especially when I don't have my computer mouse. So there's the carbon, right? So this is a 6-carbon compound, and we're making this into a 3-carbon compound, 2 3-carbon compounds. I'm not going to worry at all right now about any stoichiometry or balancing of equations. I'm just going to focus on, like I said, the essentials. So what's going on with the carbon? Where are we making energy in the form of ATP? And lastly, what's going on with those electron carriers? So ATP, we're going to make some ATP. There's going to be 2. Like I said, I'm not going to balance anything here at all. I'm just going to kind of do the basics. So to make ATP, we need ADP, and we also need inorganic phosphate. And the other thing that we're doing is we're also making NADH. So to make NADH, we are going to require NAD+. Okay, so I'm going to write a couple other notes on here, just a reminder of what's going on with these things. So ATP, this is going to go on to do cellular work. The NADH, this is going to go to the electron transport chain. And the pyruvate, that is going to go to the next step, which is the pyruvate oxidation. Okay, so hopefully these arrows and all this kind of makes sense here. Like I said, I'm kind of looking at as a reaction, over here we've got the reactants. So if you want to write it in the traditional way where you go glucose plus ADP plus PI plus NAD+, and then on the other side we have the products. So that's another way to look at it. Is reactants and products, but I like arrows, so hopefully you do too. So the next step is pyruvate oxidation. So in this case, we're going to take our pyruvate, so hopefully that's obvious that this includes pyruvate. Okay, this is a 3-carbon compound, and we're going to make acetyl-CoA. Acetyl-CoA. So this is 2 out of the 3 carbons now are left over here at this point. So what happens to the carbons? Well, we make carbon dioxide. So there's our 3 carbons. We have no ATP in this reaction, but we do have some NADH being made. So remember that takes NAD+, and there's my NADH. So remember, again, this is going to go to the electron transport chain. And not a lot of space here, but the carbon dioxide is a waste product. So I'll just put an arrow there. We've got a waste product. And that's going to be expelled by the cell. And the acetyl-CoA is going to go on to the next step. Okay, so hopefully this is pretty straightforward so far. Just a little bit of a recap, right? When you're putting these things together, don't forget all the components when you're building something. If you're building NADH, you need NAD+ as a component, right? Okay, so no ATP made there. And then last, we have the citric acid cycle, or you can call it the Krebs cycle if you like. So I'm going to draw this. I'm going to have to squeeze this in here. But we start off with acetyl-CoA. So notice in the previous reaction, I didn't even talk about the coenzyme A. So to make acetyl-CoA, you do need coenzyme A, but I'm just focusing on the basics here today. So in the end, the acetyl-CoA, that's 2 carbons, is going to make carbon dioxide. So there'll be 2 of them per acetyl-CoA, but I'm not going to balance my equations. I don't like balancing equations. Maybe that much is obvious. Okay, so there is my carbon. Okay, so where does the energy in the carbon go? The energy in the carbon goes to make ATP. So to make ATP, I need ADP and inorganic phosphate, and I'm going to make ATP. We're also going to make some NADH, so we need NAD+. We're going to make NADH. And we're going to make one more electron carrier, which is the FADH2. So in order to do that, I need FAD. So sorry for my squishing everything in here. Teaching with whiteboards is so much better, I'll just tell you that. So there's my citric acid cycle. It doesn't look much like a cycle, but like I said, it doesn't really matter at this point. I'm looking at what's going in and what are the important things coming out. And the important things coming out are ATP for cellular work, and our electron carriers for the electron transport chain. Carbon dioxide is not important anymore. It's totally a waste product, no longer essential. Okay, so let's go to the electron transport chain, and that is part of something called oxidative phosphorylation. Okay, so this is going to take a little bit more thinking about what is going on here. Okay, so in this case here, we have no carbon molecules to worry about. All the carbon dioxide is gone. Okay, so what do we have to bring into this whole thing? We have a bunch of NADHs. We have a couple of FADH2s. Okay, and so these are going to they're going to get oxidized, and we're going to get our original metabolites, so NAD+ and FADH2. Okay. And so there's one other thing that's kind of part of this equation here is we're going to have oxygen. So the oxygen is going to get, this should read FAD. And the oxygen is going to take those electrons and those hydrogens, and we're going to get water. Okay, so oxygen is our terminal electron acceptor. Terminal electron acceptor. And there's one more thing we have to add to this equation, because this is just the electron transport chain. It's not just about getting electrons and therefore chemical bonds into onto oxygen to make water. I mean, making water is great, but it's not the key thing here. And so I think the last thing we need to put is just a big arrow, is that during this process, protons are going to be pumped. And they're pumped into the intermembrane space. And we were calling this our proton motive force. So this is kind of the oxidative part, right? Here. Hopefully I have enough room to talk about the phosphorylation part. So the phosphorylation part is where we take ADP. Of course, there's no charge on that. Oops. ADP plus PI. and we're going to make ATP. And that's going to use the energy from the proton motive force. So we'll just put proton motive force there. So oxidative is the oxygen part where we are reducing oxygen. And as we're reducing oxygen, we're pumping those protons. The phosphorylation part is where we're using that proton motive force to make ATP. We're phosphorylating ADP. That's what phosphorylation means. It means we're throwing a phosphate group onto something. So that is respiration in a nutshell. Okay, so like I said, you're going to see one of these reactions either from respiration or photosynthesis, and you're going to have to be able to recognize it. And so hopefully if you know what is involved with each of these reactions, you can at least recognize it and go from there. And keep in mind where these things are going or coming from. Okay, so ATP used for cellular work. FADH2 is going to go to the electron transport chain and so on. Okay, so let's take a look here. There's kind of the overall summary of the whole thing. And you can see all those reactions there. And this is the whole point, right? We're trying to make ATP because we want to do cellular work, and that's important for cells. Okay, so let's take a little bit of a look at photosynthesis here, and then I want to kind of talk a little bit about comparing respiration and photosynthesis in kind of a way that probably you haven't seen it done before. So there's photosynthesis. Like I said, you can think of this as two reactions, is the light reactions and the Calvin cycle. So here are the light reactions. So if you want to summarize up the light reactions, you can see on the one side here we have water. So we have water, we have NADP+. So that's a little bit different from NAD+. It's a slightly different molecule. As I mentioned, it does the exact same thing, but it has an extra phosphate on it. So that's why it has the extra P on it. Okay, we have ADP, and we have PI, inorganic phosphate. So I can just get a plus for all of these things. And there's my arrow. The energy is from light, so I'll just put the light there, or I could put plus light, it doesn't really matter. Maybe I'll put that as an ingredient instead. Plus light, erase that. And then all these things, I'm just going to convert them over, right? So water becomes oxygen, NADP plus becomes NADPH, and then ADP plus inorganic phosphate becomes ATP. And those are the light reactions. So I do have a bit of an extra review of the light reactions here. I kind of want to look at them in just a little bit more detail. I think I have a slide for this, and hoping I can cram in a little bit of detail for you. I know the light reactions can be quite confusing. There's a lot of detail there and a lot of things you're not necessarily used to thinking about. So let's take a more detailed look at the light reactions. This is not the image from the textbook. This is, I think I got this in Google image searches. This is from, I think, Khan Academy. If you are looking for some good videos that explain things, by the way, I would check out the Khan Academy. Let me write that down for you. They're on YouTube, and I think they have their own website as well. And they have a whole bunch of different scientific and other concepts that are explained, and some of the videos are short, some of them are longer, and they're really well explained. It's definitely a good resource on the internet. So take a look at this. You can see there's two photosystems. You can see light. You can see all sorts of other things going on. We've got a membrane. So I kind of want to just summarize some of the important things to think about. Okay. So the first thing to think about is let's think about the light. How about we'll start there with the light. So I'm going to make a note here about light. Like I said, I'm going to try to cram things in here a little bit just to make sure I have enough room. So what can we say about the light? Okay. So the first thing about the light is we can say light provides energy. Hopefully that part is obvious. Light provides energy. And what does that energy do? Okay, energy does two things, and maybe you can see that here in this diagram here. This diagram is it's pumping protons, and it's pumping them into the thylakoids. Now, I'm just noticing that the worst thing about this photo is it's kind of upside down, I think, maybe compared to our textbook. I'm not sure. I'd have to check that. But you can see the thylakoids are right here. And so when you've got protons pumped, you're making a proton motive force, right? So hopefully that is obvious by now. So what electron transport chains are all about is making proton motive forces and ultimately making ATP. That light energy is doing something else as well. Okay, the light energy is also building new bonds. So I'm going to write that down. It's building new chemical bonds, and it's going to build them in NADPH. So I'm showing you where the bond is, right? So that's where the electrons are going to end up, is right there on the NADPH. So those electrons in that bond for that hydrogen. Okay, so that's one of the important ingredients. So now we've just mentioned bonds, and bonds are electrons. So now we have to think about the next part. Where do those electrons come from? So the electrons come from water, okay? So we can say that. We can say that water, H2O, donates electrons. So remember that reaction I showed you before. We've got water is splitting, okay? And it produces oxygen, and we love oxygen, but oxygen is just really a byproduct. So I'm gonna, like I said, kind of write tiny here. So this is a byproduct. And it's providing electrons. So those electrons, they go to the electron transport chain, and then eventually they end up on NADP+. So that's our terminal electron acceptor. That's where the electrons end up, and it becomes NADPH. And it also, the last part is some hydrogens there, and the hydrogens are ultimately just part of the solution, but some of them will be pumped across the membrane and be part of the proton motive force. So 3 things so far, right? We've got light providing energy, thinking about what that energy does, which is 2 things, pumping protons and building bonds. We've got water. Water is mostly donating electrons. We do like the fact that it gives us oxygen. That's a good thing too. And then 2 other things to mention, right, is, you know, we are always talking about our ATP and our electron carriers, right? So ATP, we can say, is produced by the proton motive force. Okay, so that was mentioned in both of these parts, the light and the water. And our electron carriers, our NADPH, is produced. So that is kind of the light reactions in terms of, you know, kind of like 90-something% of what I want you to know about the light reactions. I know there were a bunch of complicated steps. I wrote down 8 of them, I think, steps that are happening. And you know, I'm not going to ask you on the midterm to tell me all the 8 steps of the light-dependent reactions, okay? I'm trying to, you know, when I write my midterms, particularly for something like photosynthesis, I am trying to write sort of, you know, bigger picture kind of essential questions is how I see it, right? So what's important here, you know? So you could definitely see, you know, a nitty-gritty question or 2 that might ask you, what are the products of the light reactions, or something like that. You know, I want you to know these things, okay, about what is going on with all of these things. Know what NADPH is. Know what ATP is used for, right? So in this case here, right, this is the case where the answer ATP is used for cellular work is not quite right anymore, okay? Because for photosynthesis, it is used for the Calvin cycle. But most of the times when we make ATP, it is used for cellular work. So I'm going to come back to that in a minute when we compare respiration and photosynthesis. So let's take a look at the Calvin cycle. Hopefully everyone got all that. Like I said, I'm recording the lecture and I will post it on the YouTube playlist. So there's the Calvin cycle. So Calvin cycle, let's take a look. We've got ingredients. We've got carbon dioxide. We've got ATP. We've got NADPH. And I guess that's it, right? So at this point, I'm just going over here and I'm taking all these things and transforming them to their final products. So remember, our product of photosynthesis is glyceraldehyde 3-phosphate, and you can call it G3P. And hopefully if you see the full word, glyceraldehyde 3-phosphate, you'll recognize that that's just the acronym for it. ATP is going to be broken down into its parts, which is ADP plus inorganic phosphate. And the NADPH is going to be broken down into NADP plus. Okay, so what else do we need to know about the Calvin cycle? Not much. Remember, I told you an important enzyme is Rubisco. And so if you want to know the full name, that's fine. It's ribulose 1,5-bisphosphate carboxylase, but I will refer to it as Rubisco. It doesn't usually necessarily have all the capitals in there. I'm just, it's 3 words kind of put together. And that's the enzyme that grabs the carbon dioxide from the atmosphere and puts it into organic compounds. So that's the Calvin cycle. So back to photosynthesis, and like I said, what I wanted to do is spend a few minutes kind of comparing respiration and photosynthesis, really looking at some particular details. So make sure if you have any questions, you do interrupt me. That's always good to let me know what you're wanting to know. So this is the question. I think I flashed it in the lecture pretty quickly, and I said I was going to come back to it. So here we are. So you can see the question says, compare and contrast the electron transport chains and ATP synthesis in mitochondria and chloroplast. So what I wanted to do for you was put together kind of a little table for comparing things. So I started it right here. So there's the question. And you can see I'm starting off with just the basics. So it says, where are the mitochondria and the chloroplast? Okay, so mitochondria, MT is brief, short form for mitochondria. So these are found in all aerobic organisms, all aerobic eukaryotic organisms. Eukaryotic organisms. So this includes animals, hopefully that one's obvious, but plants also have mitochondria, protists, fungi. Okay. Respiration does happen in aerobic bacteria. They just don't have mitochondria to do it. They have to do a lot of the membrane stuff around their cellular membrane, so it makes it a little bit harder for them. So what about chloroplasts? Chloroplasts are found in all photosynthetic eukaryotic organisms. So what does that include? It includes plants, not animals. We got some protists, and we have algae. Okay, so remember there are photosynthetic bacteria as well. And the photosynthetic bacteria don't have chloroplasts, but they have membranes that look like the thylakoid membrane. So they're using their cellular membrane again. But I just wanted to kind of point this out, right? Mitochondria are in plants. Some people seem to forget that. And photosynthesis happens in things that are not plants. So some people seem to forget that as well. Okay, so what happens in the mitochondria? Like I said, just starting off with the basics. And so what happens there? Cellular respiration. And what happens in the chloroplast? Photosynthesis. Okay, so here's where we're going to get into the technical stuff here. And what I'm going to do is just shrink this slightly so I can cram a little bit more on the screen. Okay, so just remember mitochondria are on the left and chloroplasts are on the right. So the question says something about comparing the electron transport chains and ATP synthesis. So let's start there, okay, with the electron transport chain. So here's a question: where are the electrons from? Okay, so that's a good question, and we just talked about it in photosynthesis. Photosynthesis, the electrons are from water, right? So remember, water splits and makes oxygen, hydrogen ions, and electrons. Okay, so that one was an easy one, where the electrons come from. It's also not wrong to say the electrons come from chlorophyll, because chlorophyll does donate electrons, but they're replenished with the water. So I would type H2O, which is a little bit harder on, the, oh, hi there. Sorry, somebody was just walking in the classroom here. A little bit harder to type H2O with the subscript and all that. So where do the electrons come from in respiration? Okay, remember, electrons are found in chemical bonds. Okay, so in respiration, the electrons are from organic molecules. So we were mostly looking at glucose respiration, right? Remember that glucose is going to get oxidized, so that's where the molecule or the electrons are going to come from. What else do we have here? Can we talk about? Okay, so where do the electrons go? So where do the electrons go? And remember the word we used for this is we call this the terminal electron acceptor. So if you think about respiration, the electrons start off with glucose. We oxidize glucose, and those electrons end up on electron carriers, and those electron carriers eventually end up at the electron transport chain. They zip through the electron transport chain, and eventually they arrive at oxygen. So we can say oxygen gets reduced to water. So look at that. We have water as an ingredient of photosynthesis, and it produces oxygen. And respiration, oxygen is an ingredient, and it produces water. So there's something there. So where do the electrons go on photosynthesis? So in photosynthesis, they start off on water. Those electrons go through electron transport chain. They end up on NADPH. But the NADPH is not the final product. That goes all the way to the Calvin cycle. Then at the Calvin cycle, those electrons in the bonds end up attaching to carbon dioxide, and they end up in organic molecules. So organic molecules, more specifically, we can say G3P or glucose. Okay, so there's kind of, one way that these things are different. The electrons are we're oxidizing something in respiration, and the electrons end up in water. In photosynthesis, we're reducing something, so we're taking the electrons from water. So it's a little bit of a, kind of like I said, an opposite kind of process. Okay, so what are the electron carriers? Maybe that's another question. What are the electron carriers? Okay, so remember in respiration, we had NAD plus, and that is going to get converted to NADH. We also had FAD, and FAD gets converted to FADH2. So what about photosynthesis? We did not have NAD plus, we had NADP plus, and that is converted to NADPH. So if you do more biochemistry classes, you may see other electron carriers. There are a few out there, but these are the most common ones that you'll see out there. Okay, so what else can we talk about here? So that's kind of the electron transport chains part of it. Remember, the other part of the electron transport chains is that we're pumping protons. And so we want to make some ATP at different points. So it's worth it to talk about those things a little bit. So the next question I have is, where are the protons pumped? By protons, I mean hydrogen ions, so H plus. And another way you can say is, where is the proton motive force? So remember, in the mitochondria, where this would be the intermembrane space. And in the chloroplast, this would be the thylakoid space. Thylakoid. My mouth wants to say one thing and my brain another, and my fingers something else when I'm typing and talking at the same time. Okay, we're gonna make ATP, so that's another question. What is the ATP used for? So remember, ATP in respiration is used for cellular work. And we talked about different types of cellular work, so maybe I'll just make a little list on that. So, didn't mean for it to do that. I wanted to try that again. Well, I'll just do this with point form. So cellular work, what were we talking about cellular work? So movement of things, right? Movement of molecules across membranes. Movement of muscles, if you're an animal. What other cellular work did we talk about? Movement of organelles. And the big one, oh, I guess I'm on the next page. I'll scroll back up in a second. The big one that we talked about was energy coupling. So I'm going to put that one into bold. Okay, so let me just scroll back up and see what I can do about squeezing everything in a little bit better. Maybe what I'll do is bring these margins a little wider. And that should squeeze things in a little better. There we go, we got all that on one page, okay? So this is the big one here, energy coupling. And why do I say that's the big one? Because, you know, not everything is involving movement. And so what is energy coupling? It means driving unfavorable reactions. Unfavorable reactions. Don't like my Canadian spelling of unfavorable. So remember that means where the delta G is positive. So I think what I'm going to do, I just sort of really like this all to squeeze on one page. Just going to reduce the font size a little bit. Maybe that'll help. There we go, that is better. Okay. Yeah, and then like I said, there's lots of things that are obvious what ATP is used for when they involve movement or something like that, but building things, right? We're building organic molecules all the time. We're trying to grow, we're trying to build new cell parts, trying to do all sorts of things. So what is the ATP used for in photosynthesis? The ATP is used to fix carbon, okay? So that means take CO2 from the air and put and build it into organic compounds. So we're building things with ATP, and it's going to be G3P is the product of photosynthesis. Okay, so that's kind of my comparison, right? Of kind of looking at like I said, what I'm trying to do is compare the reactions here of photosynthesis and respiration. So if you didn't quite get all this, just let me know and I can send you this Word document that I put together. It's not a problem, just send me an email and I can do that anytime. Obviously this isn't complete, right? There are other things we could compare about photosynthesis and respiration. We could compare, you know, the locations of the reactions. We could you know, talk about you know, words that we used before. We were talking about heterotrophs and autotrophs, and you know, there's a number of other things we could look at. But in this course, these are some of the things that I care about. You know, why are we doing this? We're making ATP, and how are we doing it? We're using these electron carriers. We are oxidizing, breaking down organic compounds. Photosynthesis, in some ways, is the other end, where we're doing the opposite. We are not breaking things down, but we're building things up. And how are we doing it? We're using some of the same principles, but kind of in reverse. We're using ATP to build things rather than breaking things down to make ATP. Okay, so I'm going to go back to the PowerPoint, and I want to take a quick look at a couple of questions on that PowerPoint. And I think I just need to figure out how to get to the beginning. I'm gonna go all the way back to the beginning. This is the PowerPoint that I have on Moodle. And I just want to take a look at a few things. I'm just taking a look at the time here. I've got about 15 minutes to kind of just go over a few things on this PowerPoint. And if you tune in tomorrow, like I said, get a pen and paper, because I'm going to get, we're going to assign you some problems to look at and see how it goes with that. So like I said, just kind of a whirlwind tour through our topics and kind of remind you of a few kind of key concepts. So we had topic 7, which was the cytoskeleton. And in topic 7, we looked at the microtubules, the microfilaments, and the intermediate filaments. So you answered a question about this on the lab 4-5 lab report. Okay, so hopefully you've learned a little bit about these things, but remember they have all different functions. So microtubules, those are spindle fibers, these are flagella and cilia. Microfilaments, these are amoeboid movement and muscle movement. And the intermediate filaments really don't have anything to do with movement. They have to do with anchoring things like the nuclear lamina. So one thing that we did spend a little bit of time in the lecture was comparing the eukaryotic flagella and the bacterial flagella. So I always have some sort of question on the midterm about these things. And so hopefully you got that note, and we'll answer that question. Sometimes it's a long answer, sometimes it's shorter answer. So make sure you understand some of the differences here. Remember, there's different motions. The bacterial flagella, this thing here rotates, right? Whereas the eukaryotic flagella is more of a back and forth kind of whipping motion. They're powered by different things. The prokaryotic flagella is powered by the proton motive force, and the eukaryotic flagella is powered by ATP. So we have different systems going on. So take a look at the notes, and I drew you some nice pictures, and hopefully they can make some sense. So here's a sample multiple choice question. And if you haven't been studying, you may not know the answer to this one, but let's take a quick look. So intermediate filaments are not actin. So actin is microfilaments. Play a major role in the contraction of skeletal muscle. No, that would be actin again. Are permanent cell structures? Yes, that is nuclear lamina. Guide the movement of organelles? Nope, that would be microtubules. Are the largest? No, that would be microtubules. So the correct answer here is C. So this is what the multiple choice questions look like. You got to know your stuff, okay, and know it well. Cellular order and energetics, I'm going to kind of skip this. You can read the cartoon if you want. It's cute. Here's just a reminder about energy coupling. So you can take a look at that. Like I said, I'm just going to want to go through this relatively quickly to make sure that I have some time to address a couple of things. Here's a sample multiple choice question. So you got to know the vocabulary. What on earth is endergonic, right? So end means in, gonic means energy. So this means endergonic is bringing energy within a reaction, right? So energy is generated, not really, we're not releasing any energy. Potential energy is increased. So we are bringing things in. So that one is correct. Probably means the other ones are wrong, but let's just talk about them for a minute. Entropy is increased. Entropy means disorder. We're not doing that. We're actually decreasing entropy. We're making something complex. Okay, we just said that as well. And energy is released, which is the same as energy is generated. So again, just trying to understand the language of what is going on is going to be really important for people on this midterm. Wow, that turned out really big. Topic 9 was enzymes. So again, another unit to take a look at and review mostly the vocabulary. So vocabulary things like energy of activation. What on earth does that mean? What do I mean by inhibition of an enzyme? And lastly, we talked about phosphofructokinase as an example of an enzyme that is regulated during glycolysis. So you can guarantee there will be one question, 1 or 2 marks on phosphofructokinase. So make sure you know what it is. It's an enzyme in glycolysis. It is essential for regulation. So those are some of the key things to know. So as I mentioned, I'm probably not going to have definition questions on the midterm. I've decided to change that up a little bit. because I'm trying to make better exams for online learning, and I feel like the definitions are not really necessarily testing you properly for online exams. But there is a sample definition and a reminder for written questions that you are thorough, okay? 1 mark means probably a full sentence, maybe 2, depending on whether you make little short sentences or big long-winded ones. Examples are always good, so make sure you're thorough. 5-mark questions are going to be a lot more than just this. 5-mark questions in a written exam is a minimum of half a page. So think of that, right? Minimum. So 5 full thoughts or sentences or whatever you want to, however you want to think of it. Like I said, you don't have to have sentence form on the test, but it's not a bad idea either. So respiration. So I think let me just take a look here. I thought I had this labeled already, but I just have numbers. So we had drawn a diagram of a mitochondria, and so it's got an outer membrane, it's got an inner membrane, we've got the intermembrane space. So remember this intermembrane space, that's where the proton motive force is going to be. And we have the matrix. So you can go back and look at your notes when we drew this picture, and the matrix, of course, is where the citric acid cycle is. So TCA, the citric acid cycle. And what's number 5? Number 5 is just labeling the cristae, which is just another way to talk about the inner membrane, right? So things to know, like I said, go and review your pathways, okay? I want you to know these things about them, right? What's going on with the carbon, what's going on with the electron carriers, and what's going on with the ATP. And as a footnote, know where these things are occurring in the mitochondria or in the cytoplasm. So one thing that, it won't be a long answer question, but I'll definitely ask you some question about what happens when there's no oxygen, and the answer is usually some sort of fermentation. So remember that, glycolysis happens in the cytoplasm, no oxygen required. So we can make pyruvate and we can make ATP using fermentation, but we cannot go any further for aerobic respiration unless we have oxygen. So it shuts down all those pathways. Even though oxygen is kind of one of the last steps, it still is required for pyruvate oxidation, all that stuff shuts down due to some regulatory activities. So remember, we can make different fermentation products, and the 2 that we talked about are ethanol and lactate or lactic acid. So the advantage of oxygen is we can make a lot more ATP, right? So I think we counted, 32 ATP total, although 2 of them were from glycolysis. With fermentation, we're only getting the ATP from glycolysis, although the advantage is sometimes we're making good end products, right? Lactic acid for yogurt, ethanol and carbon dioxide for bread making and wine making. So I'm going to have some short answer questions, and I wanted to show you this one here. I think I gave you some examples last day. So some of the short answer questions are going to be pretty straightforward. So they might be something like, what happens in prophase? Some of them are going to be a little bit more technical, okay? And some of them may not have all necessarily the right answers. So I'm showing you this one because this is a hard question. And it's important to know there will be some hard questions on there. I try to find a balance of easier ones and harder ones. So that the harder ones can really reward the people that understand the material well. I'll show you an answer for this, and I'll tell you this isn't necessarily the only way to get a full mark, but this is the most correct answer, right? Why do muscle cells make lactic acid? They don't have oxygen, so that part is important. It's important to say something about not having oxygen. That would be half a mark for this question. And then the other part, it says pyruvate uses an electron acceptor to regenerate NAD+. So that is technically the chemically right answer, is that we're regenerating NAD+. So if you have that, great. That's definitely going to give you a full mark. But it's enough to say there's not enough oxygen, and therefore pyruvate is broken down only partially into lactic acid. So again, these kind of questions, the more information you give me, the better, the more likely you are going to get the full mark. Here's a long answer question that is kind of similar, and this is one actually from a previous midterm. I don't give this question. This was a previous instructor, but it's kind of, it's not too bad, but I find it's one of these questions where I'm willing to bet I would not get good responses from this one because it is technically complex, right? So, you know, it's talking about glycolysis and fermentation, and so you have to, you know, be able to talk a little bit about, you know, recycling NAD+ and all that. And at this level, I find it, it's a little bit too much. But there's probably some parts of this question you can answer, right? You know, so the first part is talking about recycling of NAD+ products of glycolysis plus fermentation. So you could talk about lactic acid and ethanol and carbon dioxide. And why can yeast live without oxygen, but you can't? Hopefully that one's obvious. You are a large, complex animal. You take millions and millions and possibly billions of ATP per second. A yeast is a very small unicellular organism. It doesn't need that much ATP, so it can just get enough ATP from fermentation. So here's the chloroplast. So we did draw a picture of a chloroplast, and so that's great. We can label this quickly. Again, we've got an outer membrane. Oops, bumped my screen. We've got an outer membrane again, and we've got an inner membrane. We have another intermembrane space, which I don't care about so much because not a lot is going on there. What we do care about is the stroma. So remember, the stroma is where the Calvin cycle occurs, and we do have these thylakoids. So sorry, I didn't plan this well enough to think about writing on the screen. That's where the light reaction occurs, and then we have the thylakoid space. So take a look at the notes I did on that because we did draw a diagram, and I made some notes about what's going on in those different locations. So the stroma is where the Calvin cycle is occurring, and the thylakoids are where the light reactions are occurring. So here we are back to this picture. Make sure that you know this, okay? Understand what is going on here. You don't need to draw it like this. Probably not going to get you to draw it, but I will ask you questions pertaining to this diagram, probably in the multiple choice section. Like I said, you know, a good question would be, what are the products of the light reactions? Or, you know, what are the reagents of the light reactions or something like that. So here's another question. So again, we have to know our words. It says all the following are associated with chemiosmosis. So you're probably thinking, okay, what is that? Okay, so that is where we are using the proton motive force to make ATP. Okay, I didn't coin the word. It's just the word that's used. So proton motive force, ATP. So yep, proton gradient, ATP being made, ATP oxidative phosphorylation. This is where ATP is being made, yes. Rubisco, no. What is Rubisco again? Rubisco is the enzyme that grabs carbon dioxide. It has nothing to do with any electron transfer chain. Doesn't even use ATP. So that one is the correct answer. So again, trying to read the question, and hopefully if you know your stuff, you'll do really well. True and false question. The purpose of the light-dependent reactions is to do what? Produce ATP and oxygen? No, unfortunately not. Oxygen is not the purpose here. I mean, we like oxygen as plants or as animals, but oxygen is the byproduct. So this should read ATP and NADPH. So that is the purpose of the light-dependent reactions. Okay, so let me just see where we are here. We had looked at that already, so I'll move on. Okay, so I just want to talk about comparing mitosis and meiosis for a minute here, and I think actually this is very similar to the table in the textbook, but it's worth kind of going through it line by line and thinking about what is going on, right? So let's compare mitosis and meiosis. Where does replication occur? Or when does replication occur? Interphase always occurs in interphase before these processes occur. You're not going to start separating DNA until it's done replicating, right? Number of divisions. So hopefully you remember that one, that mitosis has 1 division and meiosis has 2. So synapsis, what was synapsis? Synapsis is another word for crossing over. Crossing over. So unfortunately, sorry about that. There's just so many different words to refer to that process. It's hard to remember to use only the textbook words. So that doesn't happen during mitosis. There's no genetic mixing going on. It's all you're trying to get identical copies. Meiosis is where we're trying to get some genetically distinct individuals. So we're going to do some crossing over. So number of daughter cells. So in mitosis, the daughter cells divide and you get 2 daughter cells. Each of them is genetically identical to the parent cell. And if the parent cell was diploid, then you're going to have diploid daughter cells. In meiosis, you're going from diploid to haploid. And because there's 2 divisions, you're getting 2 times 2 cells, which is 4. And they're going to be genetically distinct. So what is the role of mitosis in animals? So in animals, we're talking about growth and repair. And in some, we're talking about asexual reproduction. There's a few species that do that. And what about meiosis? To produce our gametes, or eggs, and our sperm cells. Okay, so I think at this point, let me just quickly, so there's a mitosis question there. I was going to take a look at that one tomorrow. But just to give you an idea, what the wording might look like for a question, right? You can see it's talking about a cell is undergoing meiosis to produce gametes containing 2 chromosomes. So read that question. You're producing gametes to produce 2 chromosomes each. So what does that mean? It means the N number is 2. So how many chromosomes are we talking about originally? Well, we want 2N, and 2N equals 4. So make sure you can count that correctly. So I'm going to go through a problem tomorrow with everyone. Make sure you have a piece of paper available, and you can work on it, and then I'll help people once I take a look at see how they're doing. So I think that is kind of where I wanted to be for today. So hopefully, you know, you're not going to get lost following those directions there. But I do want people to do well. So what I'm going to do is I'm actually going to stop the Zoom meeting in a minute or 2 here. And what I need to do is I'm not in my office. I'm in the classroom. I'm going to actually head back to my office, but I'll restart the Zoom meeting. And anybody who wants to join in and ask me questions either about the midterm or the lab, I'll have that available and running for the next hour till about 4 o'clock. Any questions at this time? Okay, so thanks for coming out. Hopefully you got something useful out of that. I'm gonna, like I said, end the meeting now. I'll restart it up in about 3 minutes, and anybody can join in at any time if you have some questions and if you want to talk to me privately. Have a great afternoon.